Animal models of Parkinson's disease. Parkinson's disease is a neurodegenerative disorder, meaning that there is a progressive destruction in the neuronal tissue in the brain and the central nervous system. Currently, there is no cure. Parkinson's disease in particular takes hold in the substantia nigra, which is located in the ventral midbrain. The substantia nigra has important functions in motor control and learning. It acts upon these functions via dopamine, a neurotransmitter. In Parkinson's disease, there is a loss of the dopaminergic neurons, leading to a loss in the production and release of dopamine, having critical effects on the motor control. These neurons are lost due to mitochondria producing reactive oxygen species that can cause damage to the cells, leading toward cell death. The symptoms of Parkinson's disease begin in the 5th and 6th decade of life. They include a resting tremor, particularly in the fingers and hands, loss of postural reflex, poor balance, bradykinesia, a slowed movement, and poor motor coordination. As the disease progresses, the severity of these symptoms also increases. In later stage Parkinson's, towards the final stage of the disease, patients will experience hallucinations and delusions. There are currently several treatments for those at varying stages of the disease, the most common being levodopa, a naturally occurring compound that is broken down into dopamine within the brain to combat the deficit. The dopamine receptor agonists, such as pramipexole, are also in use. However, the longer that a patient is on these drugs, the more frequently the drugs start to wean themselves off and become inactive, restoring previous symptoms. Deep brain stimulation, using electrodes that are placed within the brain, attached to a pacemaker within the chest, can be used in late-stage patients to relieve some of the physical symptoms. Rotenone is a compound naturally found in the roots of some species of plant, but is used commercially in agriculture as a pesticide. Currently, rotenone is ranked as a Class 2 pesticide, meaning that it is moderately hazardous to humans. Rotenone is used clinically as an anti-mite treatment for dogs, cats, and horses. Rotenone acts by inhibiting the oxidation of NADH to NAD, which in turn blocks the oxidation of glutamate pyruvate. This leads to an increase in the reactive oxygen species being produced, which has been identified as a cause of the Parkinson's disease. Rotenone can cause these effects on the mitochondria as lipid-soluble, so it can readily cross the blood-brain barrier. Rotenone has also been identified to specifically cause this effect on dopaminergic neurons. Because of its selective actions at dopaminergic neurons, rotenone has been used to try and mimic the onset, progression, and symptoms seen in patients that are suffering with Parkinson's. By being able to reliably mimic these in animal models, new drugs and treatments can be used and tested in clinical trials and treatments. There are several different methods of administering rotenone for testing in an attempt to best mimic the Parkinson's progression. Intravenously, or IV administration, produce the specific and isolated neuron loss seen in Parkinson's disease. It was also proved to produce consistent onset of Parkinson's symptoms over several different trials on rats. Bilateral stereotactic injection involves direct injection into the substantia nigra using 3 different coordinates in surgery, allowing direct targeting. This method also was seen to produce the expected Parkinson's symptoms and neurodegenerative characteristic identified in the disease. 3 groups of male Wistar rats, aged 3, 7, and 12 to 14 months, were given doses of rotenone, 30 mg per kilogram daily, via an intraperitoneal injection. These animals also produced the symptoms associated with Parkinson's. There was also a 45% decrease in dopaminergic neurons in the substantia nigra after 2 weeks of the rats being given the daily dose. To summarize, all 3 routes of administering doses to the rats produce symptoms and degeneration that is seen within patients with Parkinson's. They all showed responding well to the medications that are currently given to patients, and there was a high reproducibility when several groups of rats were tested. This proves rotenone to be a good disease model for Parkinson's in male Wistar rats.
Methylphenyl tetrahydropyridine, also known as MPTP, was discovered as an impurity of the synthesis of desmethylprotein, a recreational drug. When 4 people were diagnosed with Parkinson's disease after having used MPTP-contaminated desmethylprotein, MPTP was able to show its effect of replicating most hallmarks of Parkinson's disease. But one of the definitive hallmarks, the Lewy body, is not replicated using only MPTP. The rat model is resistant to this toxin, but is still useful on higher mammals and mice. MPTP is a prodrug. Once it is administered, it travels through the blood-brain barrier as it is highly lipophilic. Once it is taken up by astrocytes, it is metabolized into its active form, methylphenylpyridine, also known as MPP+, by monoamine oxidase B, and is released into the extracellular space via OCT3 transporter. MPP+ then enters dopaminergic neurons via dopamine transporter. MPP+ then acts by 2 ways. One way is by being stored in mitochondrial vesicles and inhibit complex 1 of the mitochondrial electron transport chain, which will cause oxidative stress and lead to neuronal death. The second way is that there is a storage capacity in the vesicles that stores dopamine. As MPP+ is able to bind to VMAT2, the transporter that transports dopamine into the vesicles, MPP+ will push dopamine out of the vesicle into the cytosol. Dopamine will then be metabolized into toxic metabolites, such as dopal, which can be subjected to superoxide radical and hydroxyl radical attacks. The most common way of administering MPTP is by intravenous injection. When primates are treated with a high dose of MPTP, acute models will be produced. However, in recent years, new administration protocols allowed researchers to look at progressive models which would be a better replicate of pathology of Parkinson's disease patients. These progressive models allow a better study of compensatory mechanisms before the onset of symptoms. In primate models, it has been observed that the putamen has greater degeneration than the caudate nucleus. Also, there is a high variability of susceptibility of the animal towards MPTP. Normally, older animals are more susceptible. MPTP-treated animal models also respond well towards anti-Parkinson's disease treatments, such as L-DOPA and allopomorphine. They will also develop dyskinesias after long-term treatment. Overall, MPTP-treated animals are considered the standard toxin-bearing based animal model for Parkinson's disease. For example, in a recent study done by Mazinga Zhao and his research group, used MPTP-treated mice as an animal model, and was able to show that the dopamine content in the striatum after 2 or 4 treatments of MPTP will be significantly reduced after 2 weeks, whereas the substantia nigra remained at control levels. It was also shown that dopamine transporter protein expression in both substantia nigra and striatum was reduced significantly. However, VMAT2 protein expression showed no change in the substantia nigra, and only the striatum showed significant reduction in expression. This showed that MPTP treatment was able to represent acute preclinical and early clinical stages of Parkinson's disease models.
So I'm going to be looking at the 6-OHDA animal model for Parkinson's disease. So 6-OHDA stands for 6-hydroxydopamine, which is a hydroxylated analog of the natural neurotransmitter dopamine. So firstly, I'm going to be looking at some general information into the 6-OHDA animal model for Parkinson's disease. So what it does is it results in a long-lasting depletion of noradrenaline and sympathetic nerves to the heart. Today, it is one of the most common degeneration models of the central catecholaminergic projections, including the nigrostriatal system, which is particularly involved with movement. So now I'm going to be looking at some of the aims of the model. So the loss of neurons in the nigrostriatal system is one of the main pathological features of Parkinson's disease, leading to the reduction in dopamine function. The depletion of these neurons leads to motor defects, resulting in tremors and posterior imbalance, which are symptoms of Parkinson's disease. So the aim of this model is to get an insight into pathological mechanisms of the disease and to test new therapeutic targets. So now I'm going to look at some of the mechanisms of the model. So 6-OHDA accumulates in the cytosol, and this induces cell death by inhibitory actions on mitochondrial respiratory enzymes. As in Parkinson's disease, it is mainly the dopaminergic nigrostriatal pathway that is subject to degeneration. Animal models have been developed in which 6-OHDA lesions of the dopaminergic system were made. Also, by pre-treating animals with noradrenaline transporter blocker, this inhibits 6-OHDA uptake into noradrenic neurons. Administration. So 6-OHDA fails to cross the blood-brain barrier and therefore has to be injected stereotactically into the brain. Sites usually targeted are the substantia nigra, medial forebrain bundle, and the striatum. Within around 12 hours, neurons begin to degenerate, and the striatal dopamine levels are depleted within 2 to 3 days. The bilateral lesion of 6-OHDA induces symptoms as seen in Parkinson's disease. However, these animals require intensive nursing. So now I'm just going to quickly evaluate the 6-OHDA model as a whole. So one disadvantage of the model is that it does not mimic all pathological and clinical features of a patient suffering from Parkinson's disease. However, an advantage is that it induces dopaminergic neuron death while preserving other non-dopaminergic neurons. Another disadvantage of the model is that it only induces acute effects, which can differ significantly from slowly progressive Parkinson's disease.
Okay, I'm going to be talking about the PINK1 and Parkin genes, their involvement in Parkinson's, and animal models to do with those that are used to study the disease. Parkin gene was found to be the most common causative agent in early onset Parkinson's. It encodes for Parkin RBR-E3 ubiquitin protein ligase, or PARC2. And as we can see here, this is a cytosolic protein, and it's been shown to have function in targeting dysfunctional organelles for degradation via a protease. Parkin knockout mice, there have been several different kinds produced. One of the main ones I'm going to be talking about is exon 3, which shows increased striatal dopamine, decreased DAT levels, and a decreased synaptic excitability in striatal neurons. Obviously, these are many of the main brain chemistry differences in Parkinson's, so a lot of the behavioral effects are shown, especially with the synaptic excitability being decreased. This can be useful to study the unwillingness to go outside, the fright that's usually associated, and also memory loss. But the number of nigral dopaminergic neurons remains the same. We don't see the same loss that's characterized with Parkinson's. One of the main tests that can be used with these kind of mice is the object location test, is where a mouse is placed into a cage with the known objects. It's rewarded when it finds them, and it's trained to find them, and then an hour or a day later, they're placed back into the maze, and one of the items has been moved, and it will show if they've remembered the locations, and they will go to that one more for the reward. 2 impairment is another version of the Parkin KO mice. These ones actually show no behavioral effects, no catecholamine change, and actually only show age-related declines in striatal dopamine, but more importantly, an increase in D1 to D2 receptor binding. So the dopamine preferentially choosing D1 over D2, or D1 being overexpressed. Now, this is a very important hallmark in Parkinson's. Overall, these models have a mild phenotype consisting of mitochondrial dysfunction, changes in dopamine metabolism, which we were looking at, and oxidative stress. But they failed to develop the whole Parkinsonian phenotype in mice. So these knockout models provide a way to see how Parkin, the protein, is used in mitochondrial function and these different aspects, but not much else. One gene is very similar to Parkin, but it's found to be the second most causative agent for early onset Parkinson's. It encodes for PTEN-induced putative kinase 1, which is ubiquitin kinase. It's involved in protein degradation systems, but this one is localized to mitochondria. Now, the pink one knockout mice, exon 4 to 7 is going to be the one we're focusing on. This is actually produced by impairment of exon 8, which shows a knockdown effect to the other exons. They show a reduced dopamine overflow, altered dopamine signaling, and it usually combined with DJ1 or Parkin in double knockouts. One of the main aspects of this model is they show very early signs of locomotor dysfunction and very early signs of speech impairment, even though the brain chemistry remains the same. So this can be very useful for studying those symptoms to do with Parkinson's, but again, they fail to reproduce the disease as a whole. One of the main tests that's used to test motor dysfunction is the cylinder test. And during this test, a mouse is placed into a cylinder that is clear but slightly larger than itself. This actually tests asymmetry of the movements, the balance, the rearing. When a mouse is in such a small space, it's harder to move, and it actually has to place one arm up onto the curved tank and attempt to move with that. And basically, typical movements can be compared to impaired movements, and a percentage can be taken to show what's actually happened with these different mice. These can be useful for studying drugs to control those locomotor defects, like drugs to do with tremors and things like that. Pink and Parkin were actually shown to work together in regulation of mitochondria. In the 1970s, the mitochondria defects were shown to be important. And actually how this works is PINK1 accumulates on the dysfunctional mitochondria, and the kinase activity and the ubiquitin causes Parkin to translocate to the mitochondria and eventually produces an autophagosome, which can then be fused with a lysosome or a proteasome or something like that to degrade the faulty mitochondria. And in conclusion, these models fail to reproduce the main hallmarks of Parkinson's disease, in the fact that they can't reproduce the cell loss in the substantia nigra of dopaminergic cells and the presence of Lewy bodies. But they can be used to study different aspects of Parkinson's and different symptoms, such as the behavioral aspects, memory loss, the locomotor dysfunction, and they still show a lot of the altered dopamine interactions and pathways that are shown in Parkinson's, but they're not good for the entire disease as a whole.